Before I give my little intro, I just wanted to recognize the Furst family members and thank them for coming out today and continuing to support the Arthur Furst Award for faculty and alumni. It's truly appreciated. Thank you for being with us today, so thank you. I wanted to, yes. And thank you to Jeremiah Mahler, who's my assistant, who helps pretty much do everything here today. So from the food to the decorations to organizing the talk and sending out the invites, so thank you for your help with that. Nice job. So just a brief intro to let you know a little bit more about the Arthur Furst Award. It was created to honor either a USF faculty member or alumnus whose work really exemplifies the excellence in research for the betterment of humanity. And so, as I said, I'm very pleased this year that the award is going to go to Professor in Environmental Science, Gretchen Coffman. Gretchen received her BA in Biology with an ecology emphasis from Colgate University in New York. She received her MA from San Francisco State in ecology and systematic biology with an emphasis in plant and restoration ecology. And then she went from San Francisco State off to UCLA to get her PhD in environmental science with an emphasis on riparian and restoration ecology. So she came to USF in 2011, and she's been involved with both undergraduate and graduate research, focused really on ecological restoration, riparian plant ecology, and invasive plant biology in river systems of the Mediterranean and tropical climates. She's the expert leading an international effort to save the endangered cypress trees that are on the verge of extinction in Laos and Vietnam. Truly impressive, she's received external funding from the National Geographic Conservation Trust, the Chicago Board of Trade, and the University of Santa Barbara Marine Science Institute. So she has ongoing projects along rivers and watersheds in the central coast of Southern California, the San Francisco Bay Area, and the Central Valley of California, as well as Southeast Asia. Some of you might know this. In fact, when she isn't in Southeast Asia or Santa Barbara, you can usually find Gretchen off on the hillside of Lone Mountain, where she's working with the USF Landscape Department and Environmental Science students, which I think some are here today. It's great to have you. To develop a plan to restore approximately about an acre of native dune scrub that's habitat on the Lone Mountain campus. Just as a little teaser, she's going to give a tour this Friday. We have flyers on the table there, so if you can join us at 12 noon, meet us at the corner of Turk and Parker in front of the USF sign, the big USF sign. Gretchen's going to give a tour, right? And show us the amazing work you're doing, so it doesn't get better than that. In the classroom, Gretchen has a contagious energy and a clear passion for her work. She is a positive role model and a mentor for her students. This was in her application materials. One of her students wrote, that was in the MSEM, the Environmental Management Program. She wrote, Professor Coffman is truly dedicated to her students. She goes above and beyond to make sure they understand the material, often staying late in the evening to meet with them. I personally have gained so much from having her as a thesis advisor, from fieldwork in Laos to creating habitat models for an endangered tree, to learning wetland delineation. Professor Coffman has given me so many opportunities and taught me so much. The skills and knowledge she helped me develop are essential in my current position as a biologist for a small environmental consulting firm. Professor Coffman's continued work to protect an endangered tree species in Laos by working with local village groups and government officials is an excellent example of her commitment to the Jesuit values of scholarship and working towards a common good. So it gives me great pleasure, Gretchen, to present you with this year's Arthur Furst Award. Come on back.
Thank you so very much, Christina. And Dean Brooks, Dean Campari, and all those of you that helped to support this research. My family and friends, and all my colleagues in the audience that took time out from your busy teaching schedules to be here today to go through the adventure again here in this presentation, as well as I'm going to show a short film. Okay, now I'd like to talk about changing the world through my ecological restoration from Laos to Lone Mountain, with my students as a huge part of these teams. The focus of my research since I finished my PhD in 2007 has been wetland restoration for wildlife. Beginning in the Annamite Mountains in Laos, along the Nam Ton River, and adjacent national protected areas. Working with an international team of 15 wildlife biologists, I was hired right out of grad school, just finished my PhD, as the wetland restoration ecologist to survey and restore wetland ecosystems for wildlife. All right, where is Laos? Well, it's right in the middle of Indochina, where the star is right here, sandwiched in between Vietnam and Thailand and just north of Cambodia. Its population is relatively small, much smaller than the San Francisco Bay Area even, less than 6 million. Another important aspect of the sites where I was restoring wildlife habitat is that it's found along the Ho Chi Minh Trail. And yes, I'm standing here in a bomb crater. I don't know if you know this, but Laos is the most heavily bombed country in the world. Over 300,000 unexploded ordnances have been found and diffused in the national protected area in which we were working. We required UXO clearance before digging or planting anything, including our staking of our tents. Leeches and mosquitoes, however, were more of a threat than UXOs, tigers, and snakes that you may have heard about. There were many other interesting and extraordinary insects and critters like this, some of which were flying around and hitting us on the head, like this beetle on the right, lower right-hand corner. What else was interesting in this adventure was the transportation. It was a challenge, but always an adventure. We worked through not only monsoon seasons, that's plural, but typhoons as well, and had to be rescued several times. Our fearless tuk-tuk driver, however, transported us and our field care, but also our plants for the restoration, because it was so remote. We were fueled by delicious Lao food, by our top chef. Literally, this kid's name was Top. So I called him Top Chef. That consisted of sticky rice, little balls of sticky rice, and river fish, freshly caught each day, and vegetables foraged from the jungle, like bamboo shoots and ginger. The teamwork that we had was essential to the success of restoring these wetlands for wildlife, and eating meals together helped build these bonds. You can see my place at the end of this table. I got up to take the picture. All right, well, we were successful. We not only restored 30 wetlands, and these were 2-hectare wetlands, in, can you guess? 3 months. We were not allowed in the National Protected Area to use any mechanized equipment. So all we had were Lao villagers, a team of about 100 plus, and I was in charge of these people. Here's one of our wetlands and our experiment with native plants. And we were able to restore these where it would take, just for one wetland in the United States, years to restore with all the permits. This is my planting team here. What else did we do? We not only completed our goal of 30 wetlands in 3 months, but we also created salt licks for elephants, since they need to consume salts and other minerals to complement their herbivorous diets. But the most interesting, and the part that was so unexpected that I'm going to talk about right now and show a short film on, was while I was doing my field surveys, I literally tripped over what you might know of as a cypress knee, otherwise known to scientists as pneumatophores. I literally tripped over it, and I said to myself, or I thought to myself, wow, I bet this is a really unique tree up here. I looked up, it was this huge, almost 100-foot tree, and I thought, wow, this looks like the bald cypress of my Georgia childhood. I hope I get to name it. Well, it was already named, but it turns out, indeed, I had discovered a population of the critically endangered Chinese swamp cypress, the only population known to scientists in Laos. Extirpated from China, the only others known in the world were in Vietnam, and there were only 250 at the time. On my first National Geographic expedition in January to Laos, I took my graduate student Robin Hunter, who was mentioned by Christina, and worked with a team of 30 members, including international scientists, a couple of which are shown here, government foresters, shown here, villagers, and Lao University professor and a couple of their students. We had to go through 7 levels of permission. Okay? Only the first of which could be completed before our mission started, and I spent a year on that. And I've known the people I was getting the permission from for 10 years. The Lao Watershed Management Protection Authority. So we spent the first 2 weeks, which we thought we would be sampling the trees and discovering these new trees and documenting them. We spent the first 2 weeks in January of the expedition in meetings with various government officials, getting the other 6 levels of permission. And then after that, we had to go, we traveled overland, across lakes and streams and up into the mountains, and then had to meet with the village chiefs. And then sometimes it would take days to get permission from each of those, and very interesting secret meetings. Okay? But we managed to get our 7 levels of permission eventually. I'm a very persistent person, those of you that know me well. I was not going to give up before we went to the old growth forest. They said there were over 1,000 of these trees, 3 hours hiking, not on a trail, just bushwhacking up this mountain. Then we had a couple of hours to do our sampling, and then we had to head back, oftentimes with headlamps. Okay? But I didn't give up this first mission. We got to see it for 1 day. And then we got to go back in July, and we worked in it for an entire week. What else did we need? Well, I don't know if you can see in this picture the guy in the camouflage here, but this is one of our border patrols that we were required to bring with us, as well as villagers with AK-47s. You might say, this is kind of dangerous work. Well, it wasn't that dangerous. As I mentioned, the leeches and the mosquitoes were the most dangerous. What they wanted to do is they wanted to employ people, their local villagers, and the people that work in the national protected areas. So they said, we're going to come with you to protect you from the Vietnamese coming over the border and poaching this tree. And so I said, great, come along. So we had 30 people by the time it was all said and done. All right. So that's a little intro to the adventures we've had in the last few years. Now, I want to show you a short outreach film that we showed dozens of times in Laos and Vietnam this past year to help to gain the trust and permission of the villagers and the government agencies in order to survey these trees. Our producer and photographer, I want to give a shout out to right here, is photographing or filming this, so that my dad and others that couldn't be here today can see this. All right, so I'm going to show this film.
The majestic Asian swamp cypress, called Mai Hing Sam by the Lao people, was formerly widespread throughout forested wetlands of China, Vietnam, and Laos. The trees resemble a cross between coastal redwood trees of California and bald cypress trees found in swamps of the southeastern United States. In 2007, our research team identified the first stand of My Hing Sam known to science, located in a healthy wetland ecosystem on the Nakai Plateau of Laos. Most trees in the wild have been destroyed due to expanding human development. The decay-resistant wood makes the tree favorable for building houses, wooden boats, and furniture. Perhaps the greatest risk to the trees may be the clearance of wetland forest for rice paddies. The species is now listed as critically endangered by the IUCN. Until 2007, the total number of wild trees globally was approximately 250, only found in Vietnam. In January 2015, our team of researchers set out to document and help protect populations of My Hing Sam in the Nakai Nam Ton National Protected Area, located in the Annamite Mountains of central Laos. Sponsored by the Lao Wildlife Association and Watershed Management Protection Agency, our research team consisted of government officials, international scientists, local villagers, and other important stakeholders. Our team traveled overland from the capital city of Vientiane to the Nakai Plateau. We provisioned in the local markets, preparing to camp for 2 weeks in the remote mountains of the National Protected Area. Starting at the Watershed Management Protection Agency headquarters in Udom Suk, we crossed the Nam Ton Tu Reservoir, traveling upstream into the river valleys by boat and then overland by tuk-tuk, tractors, and foot. Arriving at the high village of Ban Tung, we consulted village leaders and gained permission and advice. Villagers from each town we visited joined us on our expedition to help us locate these trees. We divided up into 3 teams for our data collection: the ecology team, the collection team, and the reconnaissance team. We used GPS units to document the latitude and longitude of each tree. Using a Kestrel weather meter, we noted the elevation and weather conditions. Then, we took some basic measurements of each tree, such as diameter, breast height. We wrapped a DBH tape around the trees at 1.3 meters above the ground surface to measure their diameter directly, resulting in diameter at breast height. The height of each tree was measured using a Nikon rangefinder. If the vegetation surrounding the tree was too dense, we determined height by standing about 20 meters from the tree, measuring the distance to the tree, the angle to the top and the bottom of the tree, and then triangulating. We selected trees of various diameters from 3 stands and took tree cores from each at 1.3 meters above the ground surface. Cores were stored in labeled paper straws and taped at each end. To determine tree ages, the number of tree rings was counted back in our University of San Francisco lab. We collected cones and foliage from 80 trees in 3 stands. Cones were collected in August, October, and January to determine the timing of seed maturity. We determined that seeds mature in November from these preliminary collections. Plant tissue material was collected for genetic analyses to assist in species conservation and restoration efforts in the region. Foliage samples were analyzed by the Royal Botanical Gardens of Edinburgh to provide better understanding of the level of inbreeding and the extent of gene flow within and between the remaining stands. We determined the soil texture and color of the soil adjacent to some of the trees to understand the conditions in which they grow and that may be suitable for restoration. Soil texture was determined in the field using the ribbon method. Soil color was evaluated using a Munsell color chart, indicating the amount of organic material and other wetland soil characteristics. We measured soil moisture using a handheld TDR Field Scout. We found a total of 419 trees from 8 stands in and around the National Protected Area. The oldest tree measured was over 127 years old. However, we believe that some of the trees in the old-growth forest may be as old as 1,000 years. The largest trees were more than 3 meters in diameter at breast height and 45 meters in height. Soils were found to have a very high content of organic material and clay. Soil moisture was very high, ranging from 30 to 64% volumetric water content. Results of the genetic analysis determined that the 3 stands sampled differed significantly in their DNA composition. This means that it is essential that any restoration program should sample seeds from each of the stands in order to capture as much of the genetic diversity as possible. Healthy forested wetland ecosystems are critical for wildlife habitat, nutrient filtration, erosion control, local fisheries, flood control, carbon sequestration, and as buffers to neighboring agricultural areas. During our January expedition, we assessed the threats that these living stands face, preservation strategies, and the potential for assisted restoration of populations in the National Protected Area. Our GIS habitat suitability model indicates that there are other locations near the Ban Tung village cluster that may have Mai Hang Sam trees or be suitable for assisted restoration. Although no viable seed has been collected from swamp cypress trees in Vietnam over the last 20 years, we have successfully propagated 12 trees from the Lao population. 4 of these trees were transplanted into a wetland on the Nakai Plateau and are over 3 meters tall. We believe that more of these trees can be propagated and grown for restoration in forested areas in the National Protected Area. With our partners, we hope to document the rest of the trees in the National Protected Area and implement a protection and restoration plan supporting healthy stands of Mai Hang Sam into the future.
So again, thank you to the photographer, producer, editor, and director right here, David Blair. And grad student Robin Hunter, who was pictured there. And just a couple quick updates from summer 2015. So I returned in July of last year with Shelley Bennett, who's over here. We went back during the monsoon season. You can tell it's the rainy season here. Well, the first thing she said when out on our hike to the village where we started was, "This is my dream come true, rain in the rainy season." And I'm like, "You just jinxed us, it never stopped raining." We had to swim across the river when we were leaving after being there for 10 days. So anyway, it was quite an adventure. She's pictured here on the right next to the village chief with the hat on the right-hand side. It was really difficult that was the most difficult permission the entire 2 expeditions, but now he's part of the family. He wants us to come back and employ his villagers and help protect the Mai Hang Sam that was part of his village's spiritual forest. All right, so a couple of results. We now have found almost 600 trees during both expeditions. The 100 trees that I originally found, unfortunately, are under a reservoir now, but we found 600 more in these 2 expeditions and documented those in the ways that you saw in that film. We believe the oldest trees may be over 1,000 years old, like our red trees here in California, but we could only, the equipment we had, we could only core trees up to 80 centimeters in diameter, and the largest were over 3 meters. So we purchased new equipment and are going to head back as soon as we get a little bit more funding and go see how old those trees are and see if those old trees can tell us anything about the climate. All right, so we documented these trees, these 600 trees, and we tripled the population size that was known in the world today. The only other location they are found in the wild is Vietnam. Right here. When we visited these sites, however, this summer, Shelley and I, we noticed a stark difference in their health and appearance due to the encroachment of coffee plantations all surrounding the trees. And these trees weren't in a natural, healthy, functioning ecosystem like the ones that we had found in Laos. They were just standing there by themselves looking really awkward. They were also being flooded. Both stands were being flooded by dams that were built downstream of each of these stands. And now the population from 250 has declined to 178 or so. So it makes it even more important that we protect those healthy ecosystems and trees in Laos so we can and figure out how to propagate these trees so we can restore populations both in Laos and Vietnam. Also, the cypress were once spread, as you might think, in China. Okay, there's some dots up there in China. Well, they were only in a few locations in blue. The reason they're in red after 1960 is the only place they're left because of development of rice paddies and deforestation of all of them in the wild is in gardens, botanic gardens, and parks throughout China. So really we should call it the Asian swamp cypress. All right, so as you can see here, people are living within close proximity of these trees, next to rice paddies, and sometimes only a few in the rice paddies or next to them. So we're working with these villagers, showing them these films and identification cards, and really talking to them about the importance of protecting these trees and their significance in the world and in Southeast Asia. So we're working with them closely to develop strategies to protect these stands, and even to restore some of these stands, like this one right here. Our long-term goal, however, is restoration. We think that restoration sites can be found by using habitat suitability models, the one that Robin Hunter, my student, created that was in the film just now, and ecological data that we are collecting on each of these expeditions to best find restoration sites. The next step of this GIS modeling effort is to look at these suitable sites under different climatic scenarios, potential climatic scenarios, which David Saw is helping on with this effort. He's our director of the geospatial, am I saying that right?
It's good.
Yeah, the GIS lab, sorry. Okay, so we're also collecting cones and refining methods to propagate these trees. Okay, it's going to take them a long time to grow this large, but it's important that we figure out how to propagate them, since we were only successful in propagating to maturity 4 trees so far. Okay, those 4 trees are doing great, but we need to propagate a lot more than 4. So we're working with the Royal Botanic Gardens of Edinburgh, Philip Thomas, the IUCN conifer expert, to do this. We will involve, of course, the government, which takes a while, and all the local communities to do this restoration. Okay, we found hope in the younger stands around the National Protected Area that have many seedlings and saplings, suggesting natural recruitment and viable seed, so we can test those viable seed. So we're going to go collect from those 3 populations. We plan to return in November. Of course, I can't return in November. I'll be teaching, but I'm going to send Philip and a group over there to collect seeds for propagation. How do we collect the seeds? Well, the picture is not shown right here, but you saw a picture in the film of a tree climber. So we hire these villagers that have been doing this from childhood, collecting honey in trees. They have no equipment. They climb up these trees, 100 feet up to the canopy, way up in the canopy, and they bring down branches, which have cones and seeds. Very effective method, but what if your tree doesn't have vines for the villagers to walk or to climb up? Well, in 2007, this little 12-year-old Lao boy came with us with his slingshot and a bag of rocks. We placed large tarps around the cypress trees, and he hit the rocks up into the canopy, and the seeds and cones fell down, and we collected thousands and thousands and hundreds of thousands of these seeds. Okay? We did try to propagate all of those and only got 4 trees. So this is one of the most important focuses in our restoration program now, is to figure out how to propagate these. Let's see. So we're working with the Botanical Gardens, Pa Tat Ki, in Luang Prabang in northern Laos. We visited there in January. And we're working with the director and the botanist there to figure out how to propagate the trees and plant them. And they'll be in the new garden display when we are able to do that. And with local villages around the restoration sites shown here, we're going to have nurseries to propagate. So this is one that we created in 2007 during our Restoration for Wildlife. All right, and I just wanted to end by thanking on this Lao project. I have a couple more projects to share with you. But I wanted to end by thanking my 2 graduate students that have been invaluable team members on this research project, both in the field in these incredibly difficult working conditions and back at USF. Robin Hunter created the GIS geodatabase and conducted the habitat sustainability modeling. Shelley Bennett over here, and pictured here with me in this cave that we found, is working on Chinese swamp cypress revegetation techniques for her master's project that she's completing this spring. All right, so I want to bring you guys back to Lone Mountain at USF, where we are conducting restoration, habitat restoration that is, for butterflies like this western tiger swallowtail and other native pollinators that are really important for the city in this urban setting. Okay, recognize Lone Mountain in this picture? Okay, this is a picture from the late 1800s. It's right in the middle, just below waste. The people that were developing San Francisco thought of this as a great sand waste. 2/3 of San Francisco was a sand dune. But important dune scrub vegetation ecosystems and unique native plant and animal species lived here. What was next? Did you guys know that? 4 cemeteries around Lone Mountain were built in 1854 to 64, obviously exhumed about 100 years later, and the San Francisco College of Women was built here, which is now currently our USF's Lone Mountain campus, which was purchased in 1978. Okay, here's what it looked like before we started doing restoration. This is my first group of students on the west side of Lone Mountain. I'll show you their experiments from this year if you come on Friday. But invasive weeds, ice plant, oxalis, all sorts of fun stuff to rip out. Okay? So Professor Calloway and I have led dune restoration experiments with our undergraduate ecology labs for the past 7 years. Ecology students have developed these 3 experimental restoration plots on Lone Mountain, like this one on the west side, and there are 2 on the east side. Selecting and planting historic native plants historic to Lone Mountain. And experimental designs like this one, where they test the efficacy of weeding techniques, as well as different soil amendments. Then measuring plant survivorship and growth during the growing season. Here are my and Dr. Calloway's students from this semester. In case any of you are in the audience, each of the 3 labs One of them's going on right at this moment. This one right here. After they've planted and are so proud of their plantings, experiments, in spring 2006, we're going to monitor these through the end of the semester, and then they're going to present their research at the end of the semester. What do these experimental designs look like? Here's one of them. Through these experiments, my students are learning every step of the scientific method. Not just how to grow plants, but how to design experiments, collect the data, and then write and present the results. And applying it to native plant restoration. All right, so in my California Ecosystems upper division class in the falls, students further study dune restoration on a larger scale. This is the location of the first experiment where we planted over 400 native dune plant species in December 2012. 12 species. We now have, I think, 30, maybe even 40 species, some really rare ones too. So each fall, the class monitors these 12 species in this experimental design for their growth and survivorship in these class projects. Here are some of the beautiful plants at their best. They don't all look like this today, but many of them do. We've had a really good season, rainy season, and some sunshine lately. So again, if you want to check these out, just go over to Low Mountain at the corner of Parker and Turk behind the sign, or come join us at noon on Friday, and I'll show you what we've planted. All right, well, one other really important plant that I don't want to forget to mention is the $100,000 plant. We have 15 that we planted on campus, 13 of which have survived, and all 13 did better in this last year's drought, or the last 4 years of drought, than any other species that we planted. Really exciting news. They're even flowering now, so if you want to see them flowering, they're gorgeous. So why do they call it the $100,000 plant? They're federally endangered Franciscan manzanitas, thought to be extinct in the wild until a couple of years ago when a botanist was driving by the Doyle Drive construction site and said, that is a Franciscan manzanita, the last one in the wild. They stopped all Caltrans construction, and it cost them $100,000. There's even a video on YouTube about it. Pretty exciting for us botanists. All right, I thought I had to put in a couple of data slides just to show them a scientist, but this is a really easy one. These are all the 12 species, actually 11 species. I don't have the $100,000 plant up here, but it was 100% survivorship after this last year of drought. But we had after 3 years of drought, we had really high overall survivorship, 72%. This was data collected by my students last fall. They presented it at a conference in Southern California for undergraduate students, and we're really proud of this. Turns out 2 of the species on here that have really low or no survivorship came back amazingly well because they went dormant as seeds. Okay, California bee plant especially, and yarrow. All right, on a normal year, what is their survivorship without watering them, without irrigation? Over 90%, sometimes close to 100. That's why we're encouraging the landscaping department, hopefully, to plant more native plants in the landscape. And we've even started a USF undergraduate eco restoration club that became official this January, but it's been ongoing, maintaining the plants on campus and planting new ones. This club's mission is to promote awareness of the importance of healthy ecosystems through student involvement in restoration. And here are some of the students planting on our first work day in February. Very excited after their planting day, 2-hour planting day. I think we got over 300 plants in the ground. All right, so our club maintains the Low Mountain Native Plant Preserve, is what we call it, by weeding and planting natives each semester. But we also help local nonprofits like the San Bruno Mountain Watch with their restoration projects, like this one. This was Valentine's Day with my students this year on the top of San Bruno Mountain. And they're restoring habitat up there for butterflies, really rare butterflies. And we table, here we are tabling with the officers at different functions to talk to students and staff and faculty about the importance of native plants and ecosystems. What are we going to do next? Well, this is our planned Lone Mountain Native Plant Preserve. We're going to put signs up so that you guys know where it is, and signs next to each of the plants so you can identify them. Right now, they're QR tags that you can actually take pictures of the plants and help us monitor through citizen science as well. All right, into Marin County, I want to just mention a couple of other projects quickly before I wrap this up that my graduate students have been heavily involved in. I established a long-term riparian restoration project along Redwood Creek at Muir Beach 5 years ago. Now over 50 undergraduate and graduate students have helped me monitor the recovery of riparian restoration for coho salmon that the National Park Service has restored. This is a couple of years ago. I wanted to show the picture of the deer. For coho salmon. But what are these odd-looking shaded structures? Any guesses? You'll never guess. Coho cabanas. Coho cabanas to create shading, to cool the water temperature, and have refuges for the coho fish and other wildlife on the stream while the restoration project is recovering. So the willows were the first challenge we had. Here's our coho. You didn't know what a coho looks like? The willows were the first challenge. After the first year of planting, this is what they looked like. The National Park Service needed our help, my two graduate students and me, who did their thesis on this, to figure out why the willows were stunted. Usually they're head high or higher after 1 year. Shading by the willows, like I mentioned, is essential to maintaining ideal water temperatures for the coho salmon and other native fish. And you can't really tell here, but there was a lot of munching going on. We hypothesized that the deer had been grazing the young shoots. We also hypothesized that soil conditions, soil compaction during the restoration might be a problem. So both of my students took these two projects on for their thesis, and we first deployed remote motion and heat sensor wildlife cameras to monitor herbivory. And we found deer, lots and lots and lots of deer. 5 per day at each of the sites that we monitored, both at night and during the day. So these two graduate students and I created deer exclusion experiments. We put up, all my undergraduates and I went out for a couple of days, and Matt helped me, he's in the audience. We put up these deer exclusion experiments in an experimental design to look at if these would help during the first couple of years of restoration, and indeed, we found significantly greater height here on the y-axis is height, and second and third growing seasons on the x-axis. In black are unfenced willows, and in gray are the fenced ones. We found significantly greater height and canopy diameter, especially in that second year after restoration. Don't ask what happened in the first year. Well, I'll tell you. They had to take the fencing down before we measured because of storms that were happening. So always the challenge of working in the field. So after presenting these results to the Park Service, they began fencing all their younger willows, especially in the first and second year throughout GGNRA. All right. So what did my students do and learn? Well, they helped. They were a huge help in this monitoring. And every August, as school is starting, we go out for about 2 weeks, a team of 10 of us or so, and we monitor the vegetation for the National Park Service. And this has given my students the opportunity to learn vegetation monitoring techniques, which is really important for their career. And we have over 100 plant species they've learned how to identify. Measuring right here. Tree heights and percent cover. Right, not a bad place to work. All right, just upstream, along Redwood Creek, at Muir Woods, one of the most amazing places to work as well. I was actually asked to work on a project with a whole team of restoration scientists using dendrochronology techniques for the first time. Now I use it all the time in my research, but it was a fun time to develop this for riparian trees. Riparian trees are not usually sampled. Cores are not taken because they actually have so much water, the cores are so fragile, and they spew out like a waterfall. Water when you sample them. So it's really hard to work in these conditions. But we did it. My grad students took these samples that are shown here, worked on these 2 native plants, figured out the techniques to estimate the riparian tree age of these 2 species and understand the age of the floodplain so we can create restoration in Muir Woods. Okay? How do we do this? Well, we just simply measured the DBH of 20 of these alders here, alder trees, and then took cores and counted their annual growth rings. Turns out there's a really significant linear relationship between red alder tree diameter and annual growth rings, usually for all trees if you collect enough samples. And this equation allows us to estimate tree age non-destructively. Really important when you're working in a sensitive area like Muir Woods. Okay, so all we have to do is use this little equation here and plug in our DBHs of all the other large trees in the floodplain. Once we do that, we can figure out the tree age. And those large trees are part of a cohort that were established when large floods created those floodplains. So now we know how wide the natural meander of the stream is, and we can conduct, the National Park Service can conduct restoration in Muir Woods. All right, one more project, which is significant because I started here in Southern California. I'm continuing work that I've been doing on the Santa Clara River, where I did my PhD research on invasive arundo shown here, bamboo, tall, large bamboo-like grass species. We were just awarded a 5-year research grant to study growth of riparian plants in restoration for birds like this, leaf-bell's vireo and southwestern willow flycatcher, to create habitat for these birds that need dense willow stands. This is an exciting project for me because my students are helping me a lot and they get summer jobs out of it. And that's external funding. We have a relationship with the Marine Science Institute at UC Santa Barbara in which they're funding a much larger restoration program. So they not only get to learn restoration ecology, the science of restoration, but also the practice of restoring ecosystems. So we have a 15-acre restoration field experiment. And my 2 graduate students, Shelly and Sasha had to leave to teach my lab. But both of those guys will be there this summer. But over spring break, I was not in Southern California on the beaches. We were out there in the stream planting and measuring and setting up this 15-acre restoration field experiment, my dream project. So here's the site. We'll be planting 20 native plants over the next year under various soil moisture and ecophysiology, and shading treatments. We're going to be monitoring growth and ecophysiology of these plants. We're funded for 5 years, but I bet we'll go longer. We'll find some funding from government agencies that are interested in using this information. Believe it or not, it's really simple information, but it's not out there. And we're hoping that it will inform mitigation projects, the success criteria for those projects. So the government agencies are really excited about this project. All right, so what's next? Well, if you think this wasn't enough to manage while I have a full teaching load of 4 classes each semester, I've been planning for the last year really exciting summer class. I'll be teaching a field class in Borneo, Malaysian Borneo, with 14 master's students and undergraduate students, tropical restoration ecology. Really, really excited about this opportunity. There are a couple of the students in the audience that are as excited as me. How did this come about? Well, I've been working in the summers on a project with stewards. We're working on marine protected areas and protecting the coastline for fish and sharks and those marine ecosystems. Well, we visited this site that we just fell in love with. And we met a really passionate restoration ecologist from Australia who's been doing restoration since 1999. He's going to be vital to this project. And we'll be working with the villagers, restoring habitat on the Kinnabatongan River right here. This is degraded habitat. This is pristine reference site habitat. This river has some of the highest biodiversity of wildlife in the world, as well as the highest in Southeast Asia. So concerned about the loss of habitat due to rapid development of palm oil and illegal logging, the villagers took this into their own hands. They created a co-op to facilitate sustainable ecotourism and restoration. They developed a nursery and a planting program. We're going to help plant and propagate with the villagers. Okay? What about the science of all of this? Well, we're helping them to develop a long-term, large-scale restoration plan and experiments that will help them better conduct their restoration. That focuses on creating wildlife corridors by connecting restoration sites. We will create not only habitat, but rope bridges for orangutan that can't swim. And other species that have lost significant habitat, like this painted pitta that we photographed while hiking at night. And here are some other endemic species to this island of Borneo that we will restore habitat for. The charismatic proboscis monkey, funny looking guy. Easier to work for somebody as cute as this, our white-cheeked gibbon. Okay, that wakes us up in the morning as our alarm clock. So wildlife protection and ecosystem restoration are essential here at USF and around the world, like here in Borneo. It's essential to survival of these charismatic creatures as well as tiny little butterflies and native bees. We're not going to let the Borneo wildlife hanging, but we're going to raise money in a crowd rise for the villagers' restoration. And my TA is over here, and she's helping set up the crowd rise. And we're going to be blogging on my website, and hashtag EcoProf is my Instagram site. So we'll be having a lot of fun from the field when we do get connection to cell service. And that's it. I just wanted to thank everyone, Team the deans, my department, and department chair, Jack, unfortunately had to leave, but I wanted to thank him for nominating me. I just was blown away by receiving this about this, and it's a wonderful thing you're doing. I was really nervous to present to all of you, but I hope you learned something from it and had fun. Let's see, who else? I can't forget anyone. The Eco Restoration Club, the Master's Program, the EMBA students and the EMBS students that helped me. Craig is back there still. Thank you, Craig Peterson, Mike London, and Charlie Cross. The Landscape Department for working with us very closely on Lone Mountain to allow us to restore native plants and habitat there. And Maggie and John for being my academic mentors. And my family and friends that can't be here, they're going to see this on the video that David's doing. Thank you all. Thank you, students. You guys have any questions?
Thanks, everybody. Thank you, Gretchen.
All right, thanks, you guys.